main interest in the audience is focused ultrasound. Please keep in mind that I'm a psychiatrist, clinical psychiatrist doing research rather than a researcher doing clinical psychiatry. So I got an interest, I developed an interest in focused ultrasound basically because it's a pretty unique tool that I think the field of psychiatry has been waiting for a long time, in that it permits to produce neuromodulation. It does it reversibly, and most important, it can reach with some anatomical precision deep regions of the brain. And I think the focused ultrasound, as you would all pretty much agree, is unique in that regard, and it's a fantastic tool for clinical psychiatrists like myself who have some questions related, especially related to exploring causal relationships between the function of brain circuits and the development of symptoms, or at least basic behavioral processes that underlie depression symptoms. So I will therefore make a brief introduction related to the importance that this might have, in particular in a clinical area that is of great interest to me, which is treatment-resistant depression, which is a huge public health problem. So I will initially focus on the patient population that we have explored and we are interested in. Of course, as most of you know, major depressive disorder is among the main causes of disability in the world. And that is particularly true for persons who are in the best moments of their productive lives. And therefore, it is really a huge problem. It has worsened over the last 20 or 30 years or so. And there are some data that suggest that after the pandemic, there was a further increase in the incidence and the impact, not only human, but also financial impact of depression in this. So in just about 20 years or 10 years, the costs have been increasing remarkably. And the important thing is that this is a very common medical problem, but a very common feature of it is the lack of response to first-line treatments, including psychotherapies and antidepressant medications. So about 2/3 of people who come to the office will be prescribed one of these methods or both, and will get a significant improvement or remission. But there is about 1/3 who will start a path of treatment resistance and the need for further treatment. So, and that's an unmet need in public health in general, in psychiatry in particular. So, not surprisingly, the last 20 years or so have seen really an explosion of both established and experimental alternative treatments, maybe that can be collectively referred to as interventional psychiatry. And a big part of that is new neuromodulation methods. So there are some that are quite well established now. Treatment-resistant patients can be treated with electroconvulsive therapy, for example, that has been around for almost 100 years and remains the main the main resource we have to treat these patients. More recently, repetitive transcranial magnetic stimulation, and also some alternative medications like ketamine and esketamine. But for the most refractory of patients, there are some surgical procedures that have been developed really at the dawning of neurosurgery, functional neurosurgery, but have over the years have undergone certain refinements that now they're pretty well established. And of course, the most common and traditional interventions are those that relate to deep brain stimulation that can achieve neuromodulation in a fairly anatomically precise way, mostly for the ventral capsule, ventral striatum / anterior limb of the internal capsule. The DBS of the Brodmann area 25, or the subgenial cingulate cortex, with a lot of developments that we will not mention today. And then fairly well-established ablational procedures like capsulotomy, also targeting the anterior limb of the internal capsule, cingulotomy, mostly focusing on discrete lesions of the anterior cingulate cortex, and subcaudate tractotomy that target the most proximal and medial part of the orbitofrontal cortex. So in spite of this degree of assertiveness or assertiveness in the treatment, we only see about 50% response in patients, even in those subjected to these pretty drastic surgical procedures. And there might be different reasons for this. One, of course, is inter-individual variability in symptom dimensions. Not all patients with depression are the same. They have a huge heterogeneity in the clinical presentation, but also this is something that is pretty, I would say, fairly obvious to anatomists, but has been not quite considered until relatively recently, say the last 10 years or so. There is a huge inter-individual variability in the anatomy of circuits that presumptively underlies psychiatric symptoms of depression. So if you target always the same place, not necessarily you're going to affect the same functional circuits, and therefore that might be an additional reason that accounts for treatment non-response in these procedures. And then the question is, as you might have seen in the title of this presentation, is why we would focus on white matter tracts. I would say that there is a fairly good consensus that targeting white matter connections that participate of large-scale circuits that underlie presumptively symptoms of depression will have larger, bigger scale implications regarding neuromodulation. The reach of neuromodulation will be bigger, and that will have a better chance of symptom control. Even for targets like subgenial cingulate cortex, which initially target in the original description the limit between the gray matter and the white matter underlying it, today it's fairly clear that in order to optimize response to this method, you need to focus on which anatomical white matter tracts you are engaging, rather than the gray matter, the cortical gray matter of that region itself. So there was, as I said, I'm mostly basically a clinician, and I'm interested in that. So I think that if, the possibility of probing a circuit before a definitive intervention and being able to modulate that circuit in a reversible way, I think will open up a huge number of possibilities to optimize treatments before you get to the point of non-reversibility, either by ablation or implantation of an invasive device in the brain. Actually, this is not strange in medicine, and the cardiologists have developed a very logical pathway to achieve these objectives, and I will go back to this at the very end of my talk. So now to the more focus on the pilot study that I will talk about today. I will mention a couple of things, very basic pieces of evidence that were the rationale for us to try to target low-intensity focused ultrasound, white matter with low-intensity focused ultrasound. So there is burgeoning evidence, in vitro evidence, that ultrasound modifies the activity of myelinated axons in vertebrates. So there is some early evidence. Actually, if we go really back, the first descriptions of the effect of low-intensity focused ultrasound date back to almost 100 years ago by Harvey, and he described that the effect of focused ultrasound on myelinated peripheral nerves. So this is actually not something extremely new, but this topic remained dormant, and there was more recent interest in developing the use of focused ultrasound in white matter tracts as a potential way to achieve neuromodulation in vivo as well. So, for example, we have this relatively recent report. If we pay attention to the energies that have been used in these types of experiments, they are not logarithmic orders different from what we use in humans. So that points to translation. The other thing is that these levels of energy tend to not increase temperature significantly, pointing to some sort of alternative way in which focused ultrasound achieves its effect. And the question is that, so the main observation is that focused ultrasound in white matter tract in vitro produces a decrease in the ability of axons to transmit impulses. And going to more detailed mechanisms, Steve Broham has worked recently in understanding how potassium channels in the central nervous system are operated mechanosensitive and are operated by changes in mechanical conformation of the cell membrane. And they have actually focused on track potassium mechanosensitive channels. And he has actually characterized how membrane tension produces conformational changes in potassium channels. And this produces an increase or a decrease in the conductance to potassium across the axonal membrane. And more specifically, he has very elegantly characterized that these potassium channels are located in the nodes of Ranvier, which, as you know, are the places of the axon where the action potential is regenerated and then conducted passively to the next node of Ranvier in what is called saltatory conduction. And therefore, if you can target these nodes of Ranvier, what you might expect to achieve is a decrease in axonal conduction. This is a very nice, also recent review in which this is so the evidence, the extant evidence in this regard is summarized. And what you see here is that changes in the pressure of the membrane result in significant increases in the conductance to potassium in nodes of Ranvier, and therefore an hyperpolarization. So just to quote a recent paper by the group of Steve Broham, they predicted that ultrasound in applied to white matter would be expected to decrease spiking at the level of nodes of Ranvier, and therefore decrease the conductivity of the membrane. So with that rationale, we started our pilot study. This is a registered clinical trial. It took about 11 months for the FDA to decide it was not a significant risk study, but we finally got it, and we started about a year and a half ago, and we targeted patients with depression and varying levels of rumination, and a small sample of comparable healthy controls. I will report only on the depressive sample. We have recently finished recruitment. Therefore, these results are like really recent. And it's a single site. It's in the Laureate Institute for Brain Research, where I'm based. And lacking pretty much any sort of data for in vivo targeting of white matter tracts, we just used whatever was available in the literature at the time that we were designing the experiment. And this was a paper by the group of Robert Chen at Toronto, in which they targeted actually the motor cortex, and they demonstrated that theta burst pattern stimulation will produce changes in the plasticity of the cortex. And again, lacking any specific data that we knew of for white matter tracts, we just used the same pattern of stimulation, hoping that given the previous results I show you, in the case of white matter, what we would obtain was a phenomenon of disconnectivity or disconnection, or impairment of the transmission of action potentials through axonal membranes in humans. So this is a simplified schema of the study. This is a double-blind, randomized, sham-controlled clinical trial in which we expose participants to alternative and in a random order to either low-intensity focused ultrasound or sham stimulation. Then there is a crossover, and they will receive the other type of stimulation. We measure some clinical variables before and after sonication. This was done fairly early. We could not be sure about safety of the procedure, so we make some hypothesis testing measurements and some safety measurements. The basic hypothesis initially in the study was whether we would be able to modulate rumination acutely after a single exposure, but also there was a major feasibility. We were trying to establish feasibility by seeing whether we could see target engagement, mostly through resting state fMRI, behavioral measures, including rumination and emotional regulation. We had a self-referential task, which we haven't assessed yet the results, and then some, as I said, some safety measures. This is the setup that exists in our lab. We have the NeuroFuse Pro device and its associated BrainSight NeuroNavigator. I'm sure most of you are familiar with these devices, and we just use the transducer to apply mechanical pressure to the target. And the active, of course, it's the simple application of the transducer with a transmitted gel, and we interposed a sound-absorbing material, sorbothane, a sorbothane membrane, between the transducer and the scalp of the participant. And then we apply the ultrasound gel, and I guess it's fairly simple. And this is the pattern that we use, described by the group of Toronto, Dr. Chan's group, in which it's simple theta burst pattern of stimulation, in which you stimulate during 20 milliseconds out of 200 milliseconds, and this repeated 400 times, making a total stimulation duration of 80 seconds. For targeting, instead of, we were interested in targeting the anterior limb of the internal capsule, given the obvious clinical effects in a large number of patients with depression. But what we did instead of using a single target defined in the common anatomical space, we did probabilistic tractography in each of the participants. So we defined streamlines connecting the thalamus with both orbitofrontal and anterior cingulate cortices. And we came to an agreement between two investigators, blind to each other's choices, as to where was the highest density of streamlines connecting the thalamus with these two prefrontal structures. And this is how we defined the target. In this patient in particular, it's pretty ventral medial in the anterior limb of the internal capsule, but that we, in accordance to the extant literature, we observe a fairly important inter-individual variability. So we wanted to target individual tracts with a potential of a common function rather than a common target in defining the common anatomical space in all participants. For transducer positioning, we used, as I said, the BrainSight Neuronavigator, and this is a new incorporation. We did not have this available at the time of our study, but I just wanted to mention this is an extremely useful development using Babel Brain by Dr. Picardo, and it can be subsumed into the neuronavigation software and actually permits to image in a fairly precise way the ultrasound beam. Is a model of the ultrasound beam, but I hope it will increase even further the anatomical precision with which we can deliver energy to the target. And this is an actual picture of our lab. So let's go to the results now. The first outcome that we were trying to observe was whether this actually produced the predicted disconnection between gray matter hubs connected by the white matter tracts that we were sonicating. And indeed, it seemed to be the case. This is for 20 patients. It's a seed to whole brain connectivity analysis placing a seed in the right thalamus. And this is the contrast, the resultant contrast between sham and low intensity, an active low intensity focus ultrasound. Focus ultrasound is in gray, is in green, and then sham is in yellow. And for reference, we put the basic connectivity in the screening visit in a baseline recording outside of the stimulation protocol. And we observed, I would say fairly precisely, that we observe this connectivity, especially in the ventromedial prefrontal cortex, part of the orbitofrontal cortex. And interestingly, also the most posterior part of the cingulate cortex, extending to the precuneus. This was not something that we were predicting, but I guess would make some anatomical and physiological sense given the strong connections, white matter connections that exist throughout the cingulate bundle. So I think it's not surprising to see some disconnectivity between the thalamus and something that is more distant, like the posterior cingulate cortex. Regarding behavioral changes, we have observed non-significant decreases in negative affect and increases in positive affect variables, which, as I said, were for the most part non-significant, except mostly this one, which is joviality. And we observed that after active sonication, there was a more intense increase in joviality than after sham sonication. This is a more detailed result. And what is, this was not quite expected to us, but I think one of the ways of conceptualizing this observation is that actually in neurosurgery and in functional neurosurgery, when the anterior limb of the internal capsule is actively engaged, mirth, which is somewhat similar to joviality, it's a very common acute manifestation in the operating room related to, as I said, engagement of this target. We did not observe this clinically, but it showed up in this scale in the PANAS. On the other hand, we did not observe any antidepressant effect or, contrary to our expectation, any effect on acute effect on ruminative thinking. We also performed heart rate variability to see whether any effects in the central nervous system had it correlate in a top-down change in a top-down regulation of visceral functioning. And we use heart rate variability analysis. We also checked skin conductance level. We don't have any data for that yet. For those of you who are not entirely familiar with heart rate variability, it's a fairly simple method in which you just put in a graph what's called a tachogram, meaning the succession of RR intervals in the EKG, and that will be a signal of changes, beat-to-beat changes in heart rate variability, which can then be analyzed with a fast Fourier transform or something similar, and you see spectral components in two ranges of interest: the high frequency range, which coincides with respiratory frequency. It's a purely vagal measure, and low frequency, it reflects Meyer waves of blood pressure, which through the viral reflex impact in the heart rate variability, and therefore is a mixture of both sympathetic and vagal phenomena. So what you can have is a quotient between both these two components, and you have an approximate estimate of the sympato-parasympathetic activity onto the heart. So this is an example in one of the participants in which we obtain heart rate variability immediately before, during, and immediately after sonication. And this is the preliminary results for the initial 10 patients. It's a simple concept, but it's pretty laborious to process everything. And what we observe is that in sham, we see an increase of the sympathovagal balance, which would make some sense in that, this is a setting that produces some degree of stress. This is the way we have gotten to conceptualize what we observe here, which seems to be blunted. That increase in sympathetic over parasympathetic activity seems to be blunted in the case of those receiving, in the case of the sham condition. We have observed this in a prior study by our group, in which actually there was an autonomic changes associated with clinical changes in the case of a patient with DBS of the Brodmann area 25. And so we think that if the engagement of the autonomic nervous system is confirmed, that might be a useful tool to assess central target engagement. So to conclude the talk, what could be the main potential applications that we see to this? The first one, the one more direct, is that in order to really being able to address causation between the relationship between the function of brain circuits and behavioral phenomena, whereas neuroimaging methods can approach you to the problem, by definition, any changes in the neurophysiology in the neuroimage occur after the phenomenon of which you want to see the cause. Therefore, there is not even contingency, so you can approach it as a correlational way. Whereas if you really, this is a short but very interesting paper by Pasquale Leone and Silvanto, in which they simply show something fairly obvious, which is if you modulate a circuit as the first phenomenon, and then you observe a second behavioral or neuroimaging phenomenon, then you can approach causation more directly. And this is something that our field is needing. And I think a low-intensity focused ultrasound can be quite useful in this regard. The second potential application is, and this is very speculative, so please excuse if I go too far on this, but it gives me some enthusiasm. And I think that the story of how cardiologists have advanced knowledge in the treatment of cardiac arrhythmias can be a guiding principle to the things that we can do with these methods of reversible neuromodulation of deep structures in the brain. So typical supraventricular arrhythmias occur in a very well-characterized mechanism of reentrant stimuli in conductive tissue in the heart. It's usually located in typical places like the pulmonary veins, but that doesn't mean that it has exactly the same location in every patient. Therefore, ablating an anatomical area in the heart will not produce an improvement in all patients who could potentially benefit from the method. And I think this might be something similar to what we are seeing with neurosurgery, with the traditional approach of focusing on a specific anatomical target in all patients. So what do they do? And especially with cryoablation, which is a method that allows you to, depending on the intensity of how cold the tip of the catheter for ablating the arrhythmia is, you can usually probe the circuit first, and only when you're sure that reversibly, you're sure that you're on the target, then you produce the cryoablation. You decrease the temperature of the catheter tip to minus 80 degrees Celsius, and then you damage the tissue and you treat the arrhythmia. And this, of course, achieves over 95% of efficacy, which is much better than what we're getting in patients with treatment-resistant depression. So conceded that, of course, there are huge differences between these two excitable tissues, which are the brain and the heart. And the brain is much more complicated. There is much less understanding of the brain circuit symptom relationship. Yet, I think it might be a nice conceptual pathway to explore. High-intensity focused ultrasound used for neurosurgical ablation permits to explore this, but it's mostly to ascertain that you're in the target you want to be, rather than that you have functionally probed reversibly the relationship between the circuit that you're targeting and the symptom that you want to improve. So I think low intensity focused ultrasound, I envision that might have a role for something like this. And finally, this is the most speculative of all proposals at the time, is that we know other neuromodulation or other neuromodulation methods that, when applied repeatedly, can achieve therapeutic results. We are still far from that. In my view, we don't have very clear insights about the necessary dosing and the frequency of administration. But I would envision in a not so distant future that low intensity focused ultrasound could be explored as a therapeutic method itself to target specific brain circuits that have a role in the production of symptoms of depression. So I wanted to finish thanking all the colleagues that have been involved in this protocol, especially Dr. Akitsushi Shagaito, who has done all the experiments with me and who has taken the most responsibility for analyzing all the results I showed today. Masaya, who has helped with that. Reyes, Martin Paulus, who is my boss and the president of LIBOR. Saib, who's a dear colleague who's leaving LIBOR now, but has been part of the development of the project. Lando, who has helped, and of course, our very nice research assessment team. And you have my email and my cell phone number there in case you want to reach out to me at any point.